Hi, good afternoon, everyone. Let's give ourselves maybe another 10-15 seconds for the rest of the attendees to join us online, and then we'll begin the webinar proper. Okay, I'll start off with an introduction. Hello again, everyone, and thank you for joining us for today's APACMed webinar. My name is Gabriel. I'm the Partnership Director at APACMed. As a brief introduction, APACMed is a non-profit medical technology association representing companies operating in the medical device, diagnostics, and digital health space. As an industry association, we lead efforts to advance medical technologies in order to raise the standards of care for patients. Today, we would like to do a deep dive into the design, development, and validation of sterile barrier system. Medical devices need to be packaged in a steady, secured, and sterile environment to deliver optimum results when used by medical practitioners. Today, we are delighted to have an expert panel to share with us important consideration in sterile packaging design, development, and validation. Moderating today's dialogue is CK. CK is the Managing Director for Koch Singapore Packaging System. CK has been involved in business development through value creation in the process automation industry in Asia Pacific since 1997. As part of that journey, a part of the journey was dedicated to helping medical device manufacturers implement cost-effective material handling system. He's deeply passionate about bringing game-changing packaging automation technologies to the medical device industry. I'll leave it to CK to introduce our panelists for today. CK, it's all yours.
Warmest greetings of the day to all. Thank you very much for investing your time to join us at this webinar. Of course, thank you very much, Gabriel and APACMed, for this valuable opportunity. And today we are very privileged to have with us two expert panelists, Dr. Chang Yinxia from DuPont and Tobias Butat from Koch Park Systemic Gamberhard. We will be taking questions from the audience, and please place your questions in the chat box at the bottom of your screen. I will correct them and we will respond to you after the panelists have finished their presentations. DuPont is a very well-known brand. DuPont was founded in July 1802, more than 219 years ago. Now it's headquartered in Wilmington, Delaware, USA, and serving over 90 countries in the world. And this is a mammoth chemical industry giant. Some of the great products from DuPont include Teflon. If you cook, you know, you use the pots and pans, and Teflon is one that help you with non-stick. Kevlar, for people who are in the army, military, you use this for helmet, for protection. Nylon, and I don't have to explain anymore, nylon. Neoprene is also one of the very famous products used a lot by scuba divers. Lycra, if you're into active lifestyle, you know Lycra very well. Korean top for your kitchen top. And of course, last but not least, Tyvek. Dr. Chang's bio is in the speaker's profile, so I'm not going to expand anymore. But a little bit about Dr. Chang. She is a mother of two little boys, and when she has free time, she would like to spend time with them reading, running, and listening to the various types of music. Dr. Chang, I would like to hand the platform to you. It's all yours.
Thank you, CK, for the nice introduction about DuPont and also about myself. So let me share my screen. Good afternoon, everyone. Firstly, I want to thank APACMed, thank Gabriel and his colleagues for organizing this webinar. And I'm so honored to represent DuPont Healthcare to present here. Thank you all for joining us today. So today, my topic is important considerations in sterile packaging development. So before I formally started, let me first share our disclaimer. So this information we are going to share today corresponds to our current knowledge on the subject and may be subject to revision as new knowledge becomes available. So here is today's agenda. What we will discuss today: first, basic functions of a sterile packaging, aspects to consider in sterile packaging development, and material selection considerations, packaging design considerations, and then packaging and sealing equipment and heat sealing process development. So finally, I will touch a little bit of validation and testing of sterile packaging. Let's start by having a look of the basic functions of a sterile packaging. So actually, this is also addressed in ISO 11607. So for those who may not quite familiar with ISO 11607, this standard addresses packaging for terminally sterilized medical devices in terms of materials design and process validation. These elements are very important to be kept in mind when selecting or designing a sterile barrier system. So the sterile packaging first should allow for sterilization, which means that the packaging and the material selected should be compatible with the sterilization methods. For example, EO, gamma, E-beam, vaporized hydrogen peroxide, and so on. And second, sterile packaging should provide product protection. And actually here, there are two aspects. One, the package should provide sterile barrier, which could help hold out the microorganisms. And on the second hand, the package should provide physical protection of the device inside. And third, the sterile packaging should maintain sterility of the medical device inside till the point of use. So in order to maintain sterility, it is important to maintain the integrity of the packaging during sterilization, handling, storage, and distribution till the point of use in the healthcare facility. And also sterile packaging should allow for easy opening and aseptic presentation. This is very important because we don't want our device to be contaminated during the presentation stage. And finally, sterile packaging should provide to be able to identify the product. So in order to identify the product, sterile packaging should be able to do the printing, enabling to ensure also the traceability of the product. So now let's see the aspects to consider in sterile packaging development. So when medical device manufacturers start to design a medical device, functional team will sit down together for the design and the development. It normally involves a packaging engineer, regulatory affairs, R&D, QA, sterilization, purchasing, management, operation departments. So it's really a teamwork. So the team will consider first from the medical device itself, whether this medical device is for clinical use, whether it is a life supporting device, what is its classification, what's the competitors, distribution method, and what the materials selection, its compatibility and the safety for the patients. And packaging, sterile packaging actually is considered as part of the medical device. So the sterile packaging development is also very critical. So there are many aspects to consider. For example, the package design, whether a rigid tray should be chosen, a pouch or foam fill seal, what is the sterilization compatibility, and what is the final choice of material. So now let's move on to the material selection consideration. So in case of a packaging made of a porous material, for example, Tyvek, and a non-porous material, for example, film. So there is a need to introduce a sealant layer here. And normally there are two types of sealant on the market. First, it's adhesive sealant. And second is cohesive sealant. So as you can see here, the adhesive sealant, actually the seal and the peel function at the same point. But for cohesive sealant, it seals here, but it peels in between the sealant, inside the sealant. So one of the characteristics of cohesive sealant is that the seal is normally stronger than the adhesive sealant, and it's provide a stronger and more functional packaging design. And another characteristic, as you can see here, cohesive failure, as it's peel inside the sealant, so it normally leaves a residue, an obvious residue on the other side of the substrate, which it is removed. So this obvious residue could be a very good seal transfer and could be used as a visual inspection of the packaging integrity. Yeah, because normally the cohesive sealant is more complex in design and formulation, so it is the price is also normally higher. So as just mentioned, there is a need to introduce a sealant layer. So where this sealant layer should stay, there could be two options. The first one is that it could be coated on the porous material. And the second, it could be constructed on the film side. So on the left side, you can see for the coating on the porous material, there are different technologies like all over coating, dot coating, grid lacquer coating. And currently, many coated Tyvek, they are all over coated. And also there are some hot melt dot coated Tyvek, and its application is more in pharma and all steam sterilization. And for the multi-layer film, there are normally two technologies, for example, the co-extruded or laminated films. So one point I want to highlight here, because of the coating and also the peelable film, they may vary in formulation. So it's important to make sure that the selected coating or peelable film, they are compatible with the selected sterilization method. So from the material selection consideration, as oftentimes requested by our audience, that about the introduction of DuPont Tyvek. So here I will give a brief introduction of DuPont Tyvek. DuPont Tyvek is made of 100% virgin HDPE, and the continuous and multi-directional filaments is produced by flash spun, and the filaments is very fine. Its diameter is in the range of 0.5 to 10 micrometer, and it is bonded using only heat and pressure. So there is no binders or no chemicals involved. So it's very safe for use. And because of its unique process and the raw material used, it is very physically strong and also chemically stable. And Tyvek has been used in healthcare packaging for 49 years. It has a proven record of meeting the various industry testing requirements. And on the right side, you can see that there is the SEM picture of the surface morphology of Tyvek, and also the cross-section of Tyvek. So here I will just want to highlight a few value proposition of Tyvek properties in healthcare. So because of its unique process and property. Tyvek provides a superior microbial barrier, which is very critical for sterile packaging. And Tyvek also provides very good physical strength in terms, for example, tear or puncture. And because of its raw material is HDPE, so it's very compatible with most sterilization methods, and also it's recyclable. And its unique property and the continual filaments make clean, pure, and aseptic presentation possible for sterile packaging. Yeah, so as everyone knows that sustainability is very important to our world. So here, I'm proud to share that DuPont Tyvek healthcare grade Tyvek is 100% certified recyclable by the Institute Cyclos HTP. Because of its compatibility with different recycling techniques. And because of its lightweight and durable, so less material is actually used, and then less waste is produced. And it is with low additives and meet the rules and reach compliance. So for healthcare Tyvek grades, there are 3 core grades. The first one is Tyvek 2FS. This is ideal for foam fill seal applications for smaller devices and those with rounded edges. And Tyvek 5090B provides protection for medium risk sterile medical packaging applications and also pharma applications. Tyvek 1073B provides exceptional protection for high risk, sharp or heavy medical devices and pharma applications. So in case you are interested in or you have questions about selecting the right grade of Tyvek, you can always reach out to us and we will help you from the beginning of the product selection. So now let's move on to see the packaging design considerations. So there are many types of packaging design or configuration. For example, pure pouch, rigid tray, foam fill seal. So let's first take a look at the design considerations for pure pouch. So as you can see from this picture, there is a chevron here to facilitate the opening of the pure pouch. But you have to keep in mind, because medical personnel, they come in different size and shape. When designing this type of pure pouch, it is important to design enough room here for the medical personnel to put their hands in between to peel open the package. And also, you know, medical personnel, they always wear gloves. So with gloves on, it basically needs more room. So I actually bring 2 pouch with me here today, so I can show you on the camera. So for example, this one is with a big space here. So it's easy to get hand here, a thumb here to peel open the package. But for this one, the room is kind of small, so it's not easy to get hand here in here. And also it will affect aseptic presentation of the product inside. And another thing about peel pouch is to avoid complex folding. So for example, folding a pouch in 2 directions. This is something we want to avoid because this will create like abrasion or holes. And also folding like the pouch too tightly around internal components. And this will also cause abrasion points, not only with the inside internal components, but also with the carton outside. And this type of operation is also called scuffing. So scuffing can cause holes in package, as you can see here, SEM picture. And also, you know, films can even develop holes from flex cracking. So we hope that we can avoid complex folding while using pill pouch. So now let's take a look at the packaging design considerations for rigid tray and also form fill seal package. So first, from packaging and the product interaction perspective, we need to consider the device weight and its design. So whether we should use a rigid tray or a foam fill seal or a pouch, or whether we need to use free floating or snap fits for the medical device. And from the user interface, the sterile packaging should provide access for the easy top web removal. So for example, design a triangle here to facilitate the top web removal will be very helpful. And another thing is to facilitate the aseptic presentation and to avoid delamination and fiber tear, which is very important. As you can see here, delamination happens in this package, which is not good. It will potentially contaminate the medical device inside. So actually, in case of any type of package configuration, good peelability without fiber tear or delamination is required per usability requirement in ISO 11607. Fiber tear or delamination could generate lots of particles, and microorganisms on the surface of the package may fall onto the product together with the particles, and therefore contaminate the product. So it is very important to avoid the delamination and fiber tear of any type of packaging. So here, just showing some tips, designing tips. So for example, for the rigid tray design, it is important to avoid misplacement of the porous lid. Because when it is the lid is sealed all the way to the edge of the tray, it is easy to the fiber tear or delamination could easily happen. And also for foam fill seal, during production, there is a need to separate multiple pouch of packaging, and it is important to design some unsealed area in between and then cut. And this unsealed area is actually also called a skirt. And cutting through this unsealed area is very important because it will help to avoid delamination or fiber tear. And second, avoid oversealing. This is obvious. So with oversealing, the seal strength may increase. But with increased seal strength, it's easy to have delamination or fiber tear. And third, it is also important to replace damaged cutting equipment because a racked edge caused by a damaged cutting equipment could be a potential initiation of fiber tear or delamination. So always use sharp and undamaged cutting equipment is very important. And also lastly, a non-adapted peel system can also lead to a bad peeling behavior. So make sure that you use an appropriate sealant either on the Tyvek or on the film side to allow for good peel performance. And here is the example of some tooling that could be used to reduce like lid placement issue. So as you can see here, there are some guiding pins, which is in orange here. And the gray area is the tray flange, and the lid is in blue color. So with those guiding pins, it's easy to place the lid in the right place. So existing tool can often be modified to help with the existing tray designs. So now let's move on to the sealing part. sealing is very critical as seals are produced in the sealing process. On one hand, seals must be strong enough to withstand the rigors of sterilization, shipping, handling, and storage. But on the other hand, seals should facilitate easy access for the end user to open the sterile packaging using aseptic presentation techniques. So there are three, as we know, three primary heat sealing parameters: temperature, the dwell time, and the pressure. And these parameters are interactive. A balance between time, temperature, and pressure are needed to achieve the desired seal strength, seal integrity, visual seal transfer, and also the desired opening behavior. So from like a visual evaluation perspective, with optimum range of heat sealing process developed, sterile packaging manufacturer or medical device manufacturer will be able to get a good seal transfer with no spots or no glazing. And here you can see we also show some sealing equipments. This is a pouch sealer to seal the other side of the pouch, foam fill seal machine, and an example of tray sealing machine. So for the heat sealing process development, this is also very, very critical. And the objective is to define an operating or sealing window with validated minimum, nominal, or maximum sealing conditions. This not only result in a heat sealing process that consistently produces acceptable seals, but also can optimize energy output and operational throughput and efficiencies. So there are normally two ways recommended to optimize the heat sealing process. One is the evaluation of heat seal curves, and the other one is to use tools such as the designing of experiments using Minitab software. It really depends on the complexity of the process and the parameters. So normally the peelable film supplier or coating supplier, they will provide a heat seal curve as a reference. And this could be used as a starting point to sterile packaging manufacturer or medical device manufacturer to develop the heat seal curves on their specific machine. So here is an example of a heat sealing curve. So as you can see, with the increased temperature and the seal strength is increasing, but there is a flat area. So in this area, actually, this is a good window to produce good seal, to make seal make seals stable. It will provide good appearance, seal transfer, and also consistent strength of the sterile packaging. And another way is this is an example of designing of experiment. This is actually a 2-level 3-factorial DOE. And as we can see, the 3-factor is the temperature, time, and the pressure, and the 2-level output is the seal strength and the fiber tear or visual seal quality performance. So as you can see, those contours are seal strength, and this line are acceptable fiber tear or visual seal quality performance area. And with the overlap of the two, you will be able to define this red window, and the nominal low and high parameters will be defined in this way. Now I will talk a little bit about the validation and the testing of sterile packaging. As later on, Mr. Tobias from Co will talk more in detail about this part. So there are actually two types of validation requirements in ISO 11607, packaging for terminally sterilized medical devices. So in the first part, it's about the design validation, and the second part addresses the process validation. So for the design validation, it includes usability evaluation for aseptic presentation, which will demonstrate that the sterile contents can be aseptically removed from the sterile barrier system. And also the packaging system performance testing should prove out whether the worst case sterile barrier system can withstand the hazards of sterilization, distribution, handling, and storage. And for the stability testing, there is a real-time aging and also accelerated aging. It is to prove that the packaging could stay intact and functional over time. And normally, accelerated aging data will be sufficient enough for product commercialization. But real-time aging should started in parallel before the product commercialization. And for the process validation, for example, the sealing process validation, they normally involve like installation qualification, IQ, OQ operational qualification, PQ and performance qualification, which should prove that the process can run under statistical control and produce repeatable and making the products to meet the specification. So here, with the requirements of validation, I also list a few testing methods corresponding to these requirements. So for example, for the strength of a package closure, there is ASTM F8H seal strength test, and also there are burst strength tests. For seal integrity, we can choose from visual, dial penetration, bubble test, or combination of those. And also there are some new and emerging new packaging integrity testing methods. For example, the vacuum decay, airborne ultrasound. And for the microbial barrier, ASTM F1608 and 2630H is commonly used. So I will not go into more detail. You can always consult ISO 11607-1 2019 version, Annex B. There are more test methods listed, and the scope of use are also listed. So in summary, for a safe and reliable sterile packaging, before you start, you should understand the design and user requirements, the sterilization modality, sterilization compatibility, and the packaging materials and the design aspects is also very critical. And then the packaging equipment and the sealing process you need to understand. Then it is very important to develop a stable and wide process window. So in that case, it will be able to produce consistent products, sterile packaging that meet the requirements. And then validate your packaging process and the design is also very important. And I also want to highlight in the validation part, because in the new 29 version ISO 11607-1, usability requirement is evaluation is required. So you need to also perform that part. And then the process control measures should be established for the process. And here, I would like to share our reference used in this presentation. And you can also visit our homepage, medicalpackaging.dupont.com. And if you need any technical related documents, you may also reach out to us. Our global team of Tyvek experts is ready to assist you. And also we have a LinkedIn channel. Please feel free to follow us there. And there are always some webinar that will be posted there. And okay, yeah, that's from my side of presentation. So now I will hand over to CK.
Thank you very much, Dr. Chang, for the very comprehensive introduction. And you walk us through the material selection for leading material to ensure sterility. So thank you very much, Dr. Chang. Next, we are privileged to have my colleague, Tobias Guttert, and he's from Kopak Systeme GmbH. Kopak Systeme GmbH was established in 1969 in the Black Forest region of southwestern Germany. And I think when we mention Black Forest, the first thing that come to our mind would really be the Black Forest cake. And contrary to popular belief, not everyone from Black Forest or been to Black Forest like Black Forest cake. I've been to Black Forest many times. I don't like Black Forest cake, but I love Black Forest cake. And Cork has been, for the past 50 years, into R&D manufacturing and integration of the packaging systems for medical device, consumer, and the contact lens industry. Operating in Asia, Europe, Americas, Cork sees herself as providing a mission-critical link between the expert medical device manufacturers like you, many of you, and the hands of the surgeons that heal. Tobias, again, his bio is in the speaker profile, so I will not expand further. Just a little bit about him. Besides the time that he spent researching and understanding packaging, Tobias liked to ride his motorcycle in the Black Forest, up the hill, down the hill, and he owns a Suzuki GSX-R. He likes to visit the gym as well. Tobias, platform is yours. Thank you.
Yes. Share my screen. So. Okay, thank you, CK. I think now it's working and you can hear me. Thank you for the short introduction. Today, I want to present about important consideration, especially for the validation process for sterile packaging. And before we start, let's have a short look about the agenda. You could say today we can separate my agenda in two parts. Are you able to see my mouse? Oh, sorry, there's a pointer here. Now it should work. So we can separate my agenda in two parts. First of all, we talk in more detail about the standard, especially the well-known ISO 11607, and then also the requirements which came from the ISO 11607. And then we're going into the validation process. But we just want to talk about the basics because you could do a whole webinar just about this topic. And then we from Koch, we tried to interpret how, or want to show you how we interpret the requirements from the standard. And we collected them and called them as the quality characteristics. So let's get started with the fundamentals of the ISO 11607. So how already mentioned, it's about terminally sterilized medical devices, and it's separated in two parts. You could say on one hand, the -1 is especially for the requirements, especially for materials, such as, for example, medical films or medical grade papers, or as well Tyvek. And therefore also requirements for sterile barrier systems and some packaging system. About the details we come on the next slide. So next one in -2 there is the requirements, especially for the validation, to create such a sterile package. So this means especially the forming and the sealing, but also handling and, for example, other parts like punching or cutting. So next slide. Okay. Oh, sorry. So we mentioned the word packaging system. So the standard has a specific definition for packaging system. It's the combination from a sterile barrier system, also called SBS, and the protective packaging. About the sterile barrier system is the minimum required packaging, and I think the basic function still have been mentioned in detail on my previous from my previous speaker. And the protective packaging is defined as a material configuration for materials designed to prevent the sterile barrier system for damage. So you could say it's a really general definition. So what could it be? A protective package could be, for example, an inlay for a pouch. It could be also a carton box for a siren, and as well, for example, hard blisters with a thick film. But you could say protective packaging could be as well the upper levels of packaging level, such as, for example, a collection box of 2 sirens, or the upper level a transport box. So, I mean, well, we already seen the slide in very similar form, but I want to took the focus again and say there are some different ways where you can create some sterile packaging, and always the requirements are changed a little bit in which kind of way you create your sterile packaging. For example, the mainly known preformed pouches, they are, or reels, if you have very long sterile or medical devices. Pouches and reels are often used in hospitalities or general in facilities of healthcare. Then you also could get some preformed blisters, which are usually sized as standard, and therefore you can get some pre-cutted lid material like Tyvek lids, and can seal them as well in a let's say half-automated sealing machine. And where we want to talk today mainly about is about especially about automated form-fill sealing machine. Therefore, you can have as well hards as well as soft blisters. And just for the next slide, where we want to talk about the validation process, I shortly want to introduce you about the function of such a form-fill sealing machine. On the left top, we have a roll with the forming film. Therefore, it goes into the machine. In the machine, we usually have across the whole length chain transport with grippers. And the chain transport will transport the film during our first station. That's the forming station, where we create a cavity into the film material. This area is supposed for insert the medical device, maybe by hand or by a robot. And on the next step here, we have a second roll. This is the lid material, in this case, Tyvek, a Tyvek roll, where we insert the lid material also into the sealing station. And on the sealing station, we seal the packages. And at the end here, this is a machine for soft blisters. There's a knife where separates the sterile packaging. So now we want to talk shortly about the basics for the validation process. Therefore, I have a slide with the most common steps. So for sure, for the beginning, we have at first the design and the feasibility study. And in the first 2 steps, there are you have to consider also things of, for example, for the usability evaluation. So for example, at the design status, you always can think, how can I improve my aseptic provision? This can be done, for example, if you add at your hop blister, in this case, some gripping surfaces where even people with gloves can easily hold the package. And at the status of feasibility study, you think about, is my medical device fixed inside the package? Can it move? And if it moves, can it scratch against the film and create particles? So these are all things you have to consider in the first step. And if you think, okay, my design meets the requirements, then you will say, okay, you have to do a design freeze. If you then change something bigger, you have to justify if you have to revalidate it or not. Then we are now in an iterative process. So after design freeze, you create your first prototypes and samples, and then you have to go through different steps. Let's start with the process development. Therefore, you have to define your parameters like sealing parameters like temperature, sealing pressure, and the sealing time. And you have to do as well performance testing and stability testing. I have some separate slides next for these 3 points. And you can do them mainly simultaneously. So if you recognize, for example, at your performance testing, your peeling force is too high, then you go back to your process development and say, okay, I have to change my sealing parameters. And after changing them, you do integrity testing again. And let's say we are in the last loop and your parameters will meet the requirements and you're sure your package and will meet the requirements you defined. Then you say, before you do the parameter freeze, you create samples, do the integrity testing, and at the last step, if they pass the integrity testing, you have to do to send samples to an officially accredited laboratory. An accredited laboratory, or accredited means it's certificated from an official institution which is chosen by the government. And if your samples pass the test laboratory, you say, okay, I do the parameter freeze. Then they pass the integrity testing, and if they even pass the usability evaluation, your package is ready for the implementation to the market. So sorry about this overloaded slide, but I will summarize it for you. So we are at our first step, the process development. This means to define your parameters. Therefore, we are now at the ISO 11607, which means the validation for the packaging process. And it says in the OQ, or during the operational qualification, you have to determine your upper and lower limits for your process variables. This means in our specific case about the seal strength, you have to define an upper, in this case, or in this example, a peeling force from 2 newtons per 15 millimeter samples and 10 millimeters. And therefore you have to define your parameters. They correspond to your sealing parameters, the temperature, sealing time, and pressure. And it's very time offensive, but you have to define the upper and lower limits because, and even that, the operation or the standard ISO 11607-2 also says you have to do at least test your worst case configuration. What does means worst case configuration? It means you have to do both the test your samples from the upper and lower limit. And here you see the connection between the ISO 11607-2 and -1. It's linked because you have to do the performance testing during the operation qualification. So next step or next status I want to talk about is the integrity testing. So now we're back at the ISO 11607-1, and therefore it says to prove that your sterile packaging is kept or able to maintain the sterility, you have to do integrity testing. And this means it's separated in two parts. The standard says you have to do performance testing. That's called event related, so all physical impacts on your package, and stability testing. This means aging effects, time related things. So it also says usually the loss of sterility for your sterile barrier system is more event related than time related, but it says you have to do both. So let's start with the performance testing. There's a short overview about, you could say, the most common used test methods you can, according to the standard. There, for example, let's start here. The visual inspection, everybody will know, you just have a visual inspection of your package itself, and especially maybe with a magnifier, the sealing. Then probably everybody also will know the very famous dye penetration test, where you insert some cauterized liquid inside the package. This will visualize very small failures inside your seal. Then seal strength you could test during a peel test. Then, and some also will test the package with burst test. This means pressurize your package till it bursts. And as well bubble test, where you look for defects underwater by a wide bubble creation. And then as well, after these physical test methods, there's also a wide range of transport and simulation validations. So therefore, especially tests against vibrations, drop tests, and compression, for example, if you stack a lot of package on each other. So beside the performance testing, now we are at the stability testing. This is to prove that your sterile barrier system still can maintain the sterility just after aging. Therefore, you have two options. You can do accelerated aging with a climate chamber or environmental chamber, and real-time aging. The standard says you have to do both. You can use the results from accelerated aging to fasten up your time to market, but you have to do at the same time real-time aging. So let's have a look on this small chart I calculated. So accelerated aging is usually created by a higher temperature. In this case, the main used temperature is 55 degrees. And the most common aging aging levels are 1 year, 3 years, and 5 years. And just to show you, to reach at accelerated aging an equivalent to 1 year, you have to store them 1.3 months, for 4 months, and for 5 years, more than a half year. So this means you can really fasten up your time to market. But it also says if the real time aging results are available, for example, after 1 year, then only your results from your real-time aging are valid, and your accelerated aging results don't count anymore. And also the same for after 3 years and after 5 years. So, okay, sorry. Okay, now we're at our second part. Now I want to show you some a collection from us. We tried to show you how we to interpret the requirements. First of all, we start with packaging. These are failures where you can see easily by a visual inspection. Therefore, for example, pretty easy cracks can be easily seen by a visual inspection. Also some punctures inside the lidding material or pinholes. Then next one for sure, deformation. Your package should be free of deformations and dented edges, for example. Another one could be, in our case, especially your package should be free of crystallization. So what does it mean? For example, if an automated forming filling sealing machine stops immediately, then, for example, the film will stay under the hot sealing station, and after a while some plastics can crystallize, and this can occur in brittleness. And brittleness means if you have a physical impact, it cracks more easily. So maybe about the more interesting things, the quality characteristic we defined for, especially for the sealing seam. So this is a part already my previous speaker mentioned. We call it over sealing. It looks like transparent, or like we call it when glass is on your sealing area or on the lidding material, and this can cause some fiber tear and delamination. And yeah. The other way around is under sealing, we call it, or inhomogeneous sealing. This means your sealing areas have some unsealed spots. We also call it spottiness. And this occurs if you have in combination a too low temperature, sealing pressure, or sealing time. And we, as KOF, or recommend that the unsealed area should be not more than 10% of the whole sealing area. For sure, you can't measure this by eye. You have an optical inspection system and a software, like on the top picture here. The next type of failure will be channels. Your package should be free of channels which going through the whole sealing area, like this. And if you have sub-channels, we define, or we recommend, it should be not more than the half of the, or above the center line of the sealing area. A next type of failure could be sealing creep, it's called, or also narrowing of the sealing seam. This can happen when you, for example, expose your package during a pressure difference, like or during sterilization, or when you're if your forming process is not good enough. And we recommend not more than 15% of the whole sealing width. So we almost finish. I come to my last two slides. So we, instead of creating a sealing flange, we use a small easy trick. We say it's not allowed to punch inside the sealing area, because it can create some fiber tears. And we do a small trick. We just do a small unsealed area along the whole edge. So probably we are now at the last topic about the technical quality characteristics for the sealing. So therefore we have to go again more deep into the ISO 11607, and in chapter 5 it says some general requirements. So it says, if formed by sealing, the specified requirements for seal width and seal strength shall be met. So shall be met means you have to specify the requirements for the sealing width and the sealing strength. So let's start with the sealing width. For the sealing width, therefore, usually you're free which you define for yourself. But it's always easier, I would say, to reference to some official standards. And therefore, we have a standard, it's called the EN 868-5. And it says the total width of the seal must be less than 6 millimeters. So this means at our experience, our customers use mostly between 6 and 10 millimeters, and the most common use was 8 millimeters. And sometimes there are also pouches or ribbed seals. It also says the sum of the ribs have to be 6 millimeters. So now to the last one, the seal strength. Therefore, you have both of them. This one is a standard test method, but it don't gives you specific requirements. And if you want some specific requirements, then you can add as well the EN 868-5. And it says that after sterilization, at least you should have a sealing strength from 1.5 Newton for a sample with 15 millimeters width after steam sterilization, and 1.2 after other sterilization methods. So why is it higher than during steam sterilization? Because steam sterilization is, you could say, the sterilization method with the highest temperature stress for the package and for the sealing. So we from Koch also recommend add a little bit more than 1.5, we say, to avoid or to cover some aging effect. So we say do at least 2 newtons per 50 millimeter or even more. So yeah, now we're at the end. Thank you very much for listening and for your attention. And I give the word back to CK. Thank you.
Thank you very much, Tobias, for again very comprehensive sharing from the perspective of the heat sealing machine. Currently, we have a few questions that came in from the participants. I would like to first of all address the first question from Scipione. Sorry if I pronounce your name incorrectly, and it will be, how will a small chevron space affect aseptic presentation of product? Dr. Zhang?
Yes, so yeah, as I said, I bring some samples here today. So for example, this pouch with a smaller chevron space. So I would say there are two things. One is that with small space, it's not easy for the nurse or the doctor to find the opening, and it is not for convenient use. It's difficult to find the opening and to separate the porous material and the film and to get your hand in. So first is from a convenient use point of view. And on the other hand, because it is not easy to get in, so during you try, and there might particulates fall into this space. So when the particulates fall in this space, while you open it, the particles will fall on the device inside. So this will contaminate the device inside. But with a well designed, with enough room, you can easily find it and just you can easily find and separate and open it. So with less time spent in doing this, there is less chance of contamination.
Dr. Zhang, sorry, Dr. Zhang, I remember you mentioned that the surgeons or nurses are using these medical devices, they most of the time are in gloves. So I think with the gloves on, it will be less sensitive to be able to find when there's a very little space at the chevron to separate the lid and the pouch, or the lid and the blister. Is that correct?
Right, yeah. With the gloves on, it makes it even difficult.
Yes, and I think with the current pandemic situation, we know for sure many healthcare service providers or the healthcare professionals, they would have to deal with a lot more protection, especially it could be 2 layers of gloves instead of the usual one. So I think this point is a very key consideration in designing the packaging with the correct space. Yep. And again, sorry, back to Dr. Zhang, we have another question from Dr. Adele from Kuwait. what are the considerations or inquiries process of sterile packaging design in medical device during this COVID-19 pandemic? Dr. Chang?
Yeah, thank you, CK, and thank you for who asked this question. This is very good question. Yes, during this pandemic, we also received lots of questions about like sterile packaging design development, and also like the disinfection increase of the surface of the sterile packaging. So for example, I can share with you one increase, and also that is related with sterile packaging development considerations during this pandemic. So for example, we have customer ask us whether Tyvek package can be disinfected on the outside or using like ethanol or IPA. So actually, we don't recommend this type of disinfection, because though our customer may concern of the contamination of COVID-19 virus, but this is not really not recommended, because based on our current information, IPA and ethanol have very low surface tension and will penetrate through like a Tyvek sheet or other porous material. So this could potentially allow like microorganisms to penetrate the packaging. So that's a very big risk for the device inside. So in this case, we do recommend our customer to design their packaging, for example, to design a double or even triple entry packaging to help reduce the risk of contamination of the outside packaging surface, to control the risk of further potential contamination during the aseptic opening. So if you have concern about the outside of the surface of the contamination of COVID-19 virus, you can just simply by removing one layer and one package, and the inside should be fine. So then for the normal aseptic presentation. So that's one thing I want to share with you today. But if you have more inquiries regarding like this pandemic considerations, you may always reach out to us.
Thank you again, Dr. Chang. Staying with Dr. Adele from Kuwait. Now this question is for Tobias from Corp. During slide 8, you talk about this time as one of the factors to make sure the sealing quality is good. So Dr. Adele was asking you, how can we reduce the time consumption during the sterile packaging process, as time plays a very big role in the entire sealing process. Tobias?
Yes, thank you for the question. Therefore, I would say you still can do these test methods even if they're not validated. You can, for example, in our company, we have just picked the seal strength. We have a portable station where we have a tensile strength machine. We put them next to the manufacturing machine or the form fill sealing machine. And therefore, we create some samples, test them. And in the first, you could say in the first loop, you don't have to send your packages or samples immediately to an accredited test laboratory. You can do the tests are pretty easy. You can do them by your own. And we at COP, for example, we support our customers, and therefore we show them that our machine is capable to create such packages to meet the requirements. And let's say at the final state, if you say, okay, my sealing parameters will be fit and my tensile strength is like my reference or my requirements, then you say, okay, in the last step, I put the samples to an accredited test laboratory.
Thank you very much, Tobias. We have quite a number of questions, and this is directed to Dr. Chang once more. And this is from Julia Hong. She was asking for using the medical device packaging. microbial barrier testing according to ASTM F1608 or ASTM F2638, should they be conducted for microbial barrier testing?
Hi, CK. So the question is, let me just confirm the question. So whether the microbial barrier should be tested according to ASTM F1608 or F2638. So here the question is which one to use, or
how we can reduce the time consuming.
Should they be conducted? Either one, either F1608 or 2638. Either one, should they be conducted to prevent this microorganism from going into the sterile packaging?
Yeah, I think it's based on the risk assessment, and you can also for those sterile barrier system, sterile barrier material, porous material, you may also get the data from your supplier. For example, from DuPont, DuPont Tyvek will provide this type of data. We have a technical reference guide. We will provide those data for you, but that could be used as a reference. But it's always your decision if you want to add also that part in your validation to do a verification.
Good, thank you very much, Dr. Chang.
Both methods are acceptable.
Thank you very much, Dr. Chang. Thank you very much for participants' very nice questioning. However, we are closing to the end of the session, and I would like to thank you again for raising very, very good questions. For those we are not able to answer today, we will lock them down and we will get back to you after the session from one of the panelists. So today we are very privileged to have two technical experts to share with us this very critical part of the integrity of sterile barrier packaging, because we see ourselves as really a bridge between the expert medical device manufacturers and the hands that heals, our surgeons. So today, again, we thank you, Dr. Chang and Tobias, for sharing with us your technical expertise. I would like to hand the platform back to Gabriel, who will share with you a little bit to close the session.
Yep.
Thank you very much.
Thank you, CK, for the very, very nice closing. I think you summed it very well. There are lots of key takeaway, we learned so much that, the design, development, and validation of a sterile barrier system is of great importance to any medical device manufacturer. I also would like to thank Dr. Chang and Tobias for your insightful, presentation. Before we go, I'd just like to also extend an invitation to you for you to join us at APAC Med's upcoming Asia Pacific MedTech Forum, scheduled for 19th and 20th of October. So this is APAC Med's flagship event happening next month. We invite you to join us to hear from an impressive lineup of speakers, sharing their vision around our theme, which is Health Future by 2025, and also having the opportunity to experience a virtual showcase highlighting the latest medical innovation over these 2 days. So thank you once again to all of you for joining us. Everybody stay safe, stay healthy, and we'll talk again soon. Thank you very much and have a good evening. Bye-bye.
Bye-bye.
Thank you. Bye-bye. Thank you all for your attention, for your time.